The software returned an age for the mutation, and the number was wrong. Not wrong in the way a rounding error is wrong, wrong in the way that suggests the model itself has misunderstood something fundamental. The researchers reran the calculation, same number, 20 million years. For a variant sitting quietly inside a single human gene, that number made no sense. Humans hadn't existed for 20 million years. Neither had most of the species being compared against. So either the software was broken, or the gene itself had been carried unchanged through an evolutionary lineage stretching back before there were hands to hold a test tube, before there was a species called Homo sapiens to study it. The gene in question sits on chromosome 9. It decides something most people only think about once, the moment before a transfusion, on a form in a single letter, A, B, AB, O. That letter turns out to be one of the oldest continuous decisions biology has ever made and refused to unmake. For most of the last century, blood type has occupied a strange, almost bureaucratic corner of the popular imagination. It shows up on a hospital wristband. It gets asked once, memorized rarely, and otherwise ignored, treated as background information, useful only in an emergency, unconnected to anything larger about who a person is or where they come from. In parts of East Asia especially, a popular belief took hold in the 20th century that blood type shapes personality, that type A people are careful and anxious, type B people are creative and unpredictable, type O people are confident natural leaders. A related idea, the so-called blood type diet, claimed that each type reflects a different point in human dietary history, and that eating according to type could improve health. Neither claim has held up well under scientific scrutiny. Researchers who have tested these ideas directly have found no reliable link between ABO blood type and personality traits, and no consistent evidence that a blood type-specific diet produces better health outcomes than any other approach. These are ideas people have believed sincerely, and for generations, without them turning out to be true. The actual history of the discovery is almost as ordinary as the myths built on top of it. In 1900 and 1901, an Austrian physician named Karl Landsteiner noticed that mixing blood from different people sometimes caused it to clump, a discovery that eventually won him a Nobel Prize and made modern transfusion medicine possible. For decades afterward, ABO blood type was treated as exactly what Landsteiner found it to be, a practical medical classification, useful at the moment of a transfusion and largely irrelevant the rest of the time. No one in that laboratory in 1901 had any reason to suspect they were looking at something tens of millions of years old. The actual biology is simpler and stranger than either myth. The ABO gene sits on chromosome 9, and it does one narrow job. It tells the body which sugar molecules to attach to the surface of red blood cells. Type A cells carry one sugar flag. Type B cells carry a different one. Type AB cells carry both. Type O cells carry neither, a blank surface, a flag never raised. Think of it less like a bloodline and more like a uniform patch sewn onto a cell's outer layer, invisible to the eye but instantly recognizable to the immune system, which is exactly why mismatched blood types trigger a violent rejection during a transfusion. That is the entire textbook explanation. A gene, a sugar, a flag. Nothing dramatic. Nothing that should have drawn the attention of evolutionary biologists studying primates tens of millions of years removed from human history. Here is where it gets complicated. When geneticists began comparing this supposedly minor gene across species, not just among humans, but across apes, monkeys, and distant primate relatives, the gene refused to behave the way an ordinary, unremarkable trait should. Ordinary traits drift. They mutate, disappear, get replaced, following the slow, random churn of evolution. This one didn't. It kept showing up, generation after generation, species after species, in a pattern that looked less like coincidence and more like inheritance. The anomaly is this. The A and B versions of this gene are not unique to humans. They appear nearly identical at the molecular level in gibbons, in orangutans, in macaques, baboons, and colobus monkeys, species that split from the human lineage tens of millions of years ago, long before anything recognizable as a human being existed. A 2012 study published in the Proceedings of the National Academy of Sciences, led by geneticist Laure Segurel, examined exactly this pattern across primate species and reached a conclusion the researchers themselves called remarkable. The A and B blood types are not the product of separate species independently evolving the same trait by coincidence. They are what geneticists call a trans-species polymorphism, a genetic variation so old it was already present in a common ancestor, before that ancestor's descendants ever split into the separate species we know today. Think of it like an heirloom passed down not through one family, but somehow inherited identically by several unrelated families who share only a distant relative none of them ever met. Crucially, the researchers found this wasn't a case of the same mutation happening to occur twice by chance. The underlying genetic sequence matched too closely in too many places for coincidence to explain it statistically. The variation had to have been inherited, whole, from a shared ancestor, then carried forward separately down every one of those diverging branches. The study placed the age of this shared variation at approximately 20 million years. A separate line of molecular research, comparing ABO sequences across humans, chimpanzees, gorillas, and orangutans, put a conservative floor on the same variation of at least 13 million years, meaning even the cautious estimate reaches back roughly 60 times further than the entire span of recorded human history. Here is why that number breaks the model researchers expect. Ordinary genetic variation, the kind with no particular survival advantage attached to it, does not survive that long. Left alone, undefended by selection, a genetic variant gets eroded by random drift, overwritten, lost, usually within a few million years at most. For a variation to persist for 13 to 20 million years, something has to be actively defending it, every generation, across the boundary separating one species from another. Scientists call this mechanism balancing selection, a kind of biological standoff, where no single version of the gene is ever allowed to fully win, because each version carries its own advantage against a different threat, keeping the whole system locked in permanent, deliberate tension. Not by accident, if evolution can be said to design anything at all. Not by years, by tens of millions of years. That is not a footnote on a hospital form. That is one of the oldest, continuously defended features anywhere in primate biology, and researchers are still not entirely certain what has been worth defending it against for so long. The first theory, and the one most widely accepted among researchers, is pathogen resistance. The idea is that different blood types offer different degrees of protection against different infections, so no single type can ever fully outcompete the others, because whichever type is currently disadvantaged against one disease is often protected against another. It is a kind of evolutionary tug-of-war, and it explains, in principle, why the variation never resolves into a single winner. There is unsettlingly current evidence for this. A 2021 laboratory study published in the journal Blood Advances, conducted by researchers affiliated with the American Society of Hematology, found that the receptor-binding protein on the surface of the SARS-CoV-2 virus, the mechanism it uses to attach itself to human cells, binds preferentially to the sugar flag carried by type A blood cells. A separate 2021 case control study of healthcare workers found that people with type A or type AB blood faced a 36% higher chance of developing severe COVID-19 than those with type B or O, while type O carried a measurably protective effect. The numbers were specific enough to publish. They were also, researchers were careful to note, probability, not certainty, a statistical tilt, not a guarantee. Here is the limitation. If pathogen resistance fully explained the pattern, the effect should show up consistently, everywhere researchers looked for it. It didn't. Early reports out of China first flagged type A as a susceptibility risk, and studies from Italy and Spain reported the same pattern for severity specifically. A large Danish study, working from a different population entirely, found a link to susceptibility, but not to how severely the disease progressed once contracted. Then a separate American study, examining tens of thousands of patients in Boston and New York, found no significant link at all between blood type and COVID-19 severity. The effect that seemed unmistakable in one population simply vanished in another. 4 countries, 4 different answers, using roughly the same question. Pathogen resistance explains why the variation persists across deep time. It does not explain why the same virus, the same protein, the same underlying biology produces a measurable effect in one study and no effect at all in the next. The second theory is more complicated because it adds a second variable most people never think about at all, the Rh factor, the positive or negative sitting quietly next to the letter on a blood type. A haplogroup in genetics functions something like a surname passed down not on paper but inside the body itself, a marker of shared ancestry running through the DNA rather than through a family tree. Rh status works a little differently, but the analogy still holds in one sense. It is another layer of inherited identity, stacked directly on top of the ABO type. And when researchers began accounting for both factors together instead of separately, the picture changed. A 2020 case control study conducted in Kunduz, Afghanistan, examined COVID-19 patients against a control group of more than 1,000 blood donors, tracking ABO and Rh status simultaneously rather than in isolation. The combination of type A blood with a negative Rh factor was associated with a risk increase researchers measured at more than 7 times higher than baseline, a number dramatically larger than either factor produced on its own. Something about the interaction between the two systems appeared to compound the risk in a way neither system explained alone. Researchers proposed possible explanations: differences in how antibodies form, differences in inflammation response, differences too subtle to isolate with the sample size available. But none of these explanations has been confirmed. The interaction is documented. The mechanism behind it is not. This is the part the records don't fully explain. The study came from a single regional population, a few hundred cases, one hospital. Larger studies conducted afterward in Boston and New York, working with far bigger patient populations, failed to reproduce any meaningful link between ABO blood type and COVID-19 outcomes at all. The data doesn't confirm the Rh-ABO interaction is real everywhere, it only fails to rule it out. The third theory, and the one many people find hardest to let go of, is the popular claim that Rh-negative blood originated with the Neanderthals, passed into modern humans through ancient interbreeding, a kind of leftover signal from a vanished species. It is a compelling story. It is also, as far as the ancient DNA can show, incorrect. When researchers sequenced the genomes of Denisovan and Neanderthal individuals recovered from Siberia and Spain, they found both were homozygous for the Rh-positive variant, meaning neither carried even a single hidden copy of the negative gene. Whatever the ultimate origin of Rh-negative blood turns out to be, it did not come through this particular genetic doorway. But the actual finding is stranger than the myth it replaces. A 2021 study from a research team affiliated with Aix-Marseille University, published in the journal PLOS One, sequenced the blood group systems of 3 Neanderthal individuals and 1 Denisovan, and found something modern humans do not carry at all: a unique Rh allele, absent from almost the entire modern human genome, with exactly 2 documented exceptions, 1 Aboriginal Australian individual and 1 Papuan individual. Researchers connected this allele system to a heightened risk of hemolytic disease between mother and fetus in cases of Neanderthal-human interbreeding, raising the possibility, treated cautiously and without resolution, that blood incompatibility itself may have quietly limited how successfully the 2 populations could interbreed. Researchers are careful to frame this as a contributing factor, not a solved cause of the Neanderthal disappearance. The data doesn't explain why that allele survived in exactly 2 people out of billions on opposite sides of the Pacific. There is a related puzzle sitting underneath all of this, one researchers have wrestled with separately for decades. Rh incompatibility between a mother and her fetus can be dangerous, sometimes fatal, in cases where an Rh-negative parent carries an Rh-positive child. In theory, natural selection should have quietly eliminated a gene that carries that kind of cost. It hasn't. Rh-negative blood remains common enough in some regions that the mechanism defending it has to be doing something to offset the danger, some protective advantage strong enough to outweigh the risk to a pregnancy. Researchers are careful to frame this as probability, not certainty. No single explanation has been confirmed. The gene survives. The reason it survives is still, formally, an open question. Picture a rock shelter somewhere in Ice Age Europe, 25,000 years before any researcher ever ran a single sequence. The air sits just below freezing. Animal fat burns in a shallow stone lamp, throwing orange light across a wall already marked by older hands. The smoke smells of tallow and wet stone. Someone coughs, low, in the dark past the firelight. A small group shares meat from that day's hunt, passing it hand to hand in the smoke and the cold, none of them aware of anything happening inside their own bodies. Somewhere in the blood moving through their veins, a molecular decision made tens of millions of years earlier is still being carried forward, a decision older than this cave, older than fire, older than the species crouched around it. They cannot see it. They will never know it exists. Outside, wind moves across ice that will not fully melt for another 10,000 years. The sugar flag on their cells will outlive every one of them, passed forward through births and deaths and migrations they cannot imagine, into descendants scattered across every continent, still carrying the same signature, still defended by the same invisible tug-of-war between resistance and vulnerability that has run unbroken since long before there were hands to hold a torch. The question was already inside them. It still hasn't been answered. Here is the twist the research keeps circling back to, without ever quite landing on it. The same trait that makes type A blood measurably more vulnerable to one 21st century virus is, at the molecular level, the same category of trait that has been protecting humanity, and its primate relatives, from other pathogens for tens of millions of years. Vulnerability in one century, survival tool across geological time. Both true. Neither cancels the other out. There is a second layer underneath that one, and it cuts against something people like to believe about blood type. In forums and family group chats, blood type sometimes gets folded into a story about ancestral purity. The idea that a rare type marks a bloodline as somehow distinct, older, more original than the rest. The genetics says almost the opposite. The A allele survived in humans, it survived in gibbons, it survived in orangutans, macaques, baboons, colobus monkeys, none of them descended from one another, none of them in contact, all of them carrying a piece of the same inheritance from a common ancestor none of them ever met. Not a bloodline, not a nation, not a chosen lineage passed down through one people alone. Shared, across species, long before there were peoples to divide it among. A geneticist who has spent years studying primate blood systems described the finding plainly in an interview after the trans-species polymorphism results were published. The team had expected, going in, that a trait this specific would turn out to be a case of separate species, independently landing on the same solution by chance. What they found instead was inheritance, a shared thread, not a coincidence. What feels, on a hospital form, like the most personal piece of information a person carries, turns out to be one of the least personal things about them at all. A multi-species heirloom, worn by nearly everyone, understood by almost no one. Somewhere, right now, someone is looking at their own lab results, seeing those two characters, A, positive, and reading them as a small, closed fact about themselves. A single line on a form, nothing more to think about. That reading is not wrong exactly. It is only incomplete, in a way most people will never have reason to discover. Zoom out far enough, and type A blood stops being a story about one letter on a form. It becomes a case study in something larger. The way ordinary-looking traits, the ones people barely register on a piece of paper, sometimes encode the oldest and most consequential battles in the history of life. Every blood type carries some version of this same deep argument between resistance and exposure, written in a code that predates the species now arguing about it. Type A is simply the version currently under the microscope, the version a virus in this century happened to notice first. Researchers are careful to say this is probability, not certainty, and the picture keeps shifting as new studies land, sometimes agreeing, sometimes flatly contradicting the ones that came before. That is not a failure of the science. That is what an unfinished argument looks like from the inside. The sugar flag raised on the surface of a red blood cell was planted there by something that stopped being human or ape or monkey tens of millions of years before any of those categories existed, and it is still standing, still being defended, in blood moving through veins right now, in people who have never once thought about where it came from. If you have type A positive blood, DNA has not finished revealing the truth. It has only shown how much older the question is than anyone assumed.